All right, welcome everyone. Thank you for joining us today. It's time to get started. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix. Today, Gabe Rudy, our VP of Product and Engineering, will present the newest addition to the VarSeq software suite, VS Reports. VS Reports takes the output of your tertiary analysis to a customized clinical report in just one click. Gabe, I know you're excited, so I'll pass the mic to you.
All right, well, thanks, Cheryl. And thanks again for joining us today. So to give you a quick outline of what we'll be covering, in very short time, I'd like to actually give you a feel of what VS Reports looks like. So what VarSeq looks like in this clinical setting. From the context of if I was a lab tech, MD, pathologist, and I was using VarSeq to do my job, what does that final step look like? To do that, I want to give you a little bit of context about Golden Helix and NGS testing, the kind of clinical gene testing that we'll be talking about today, and the background and what the current kind of workflows are. And then we'll jump into a very quickly we'll be able to get into a demonstration of VS Reports in that context of the person doing the work. Once we have that context in place, we can step back and talk about VarSeq as a clinical NGS platform and the components that go into making it so that you can do the R&D step, prepare your test, and put it into production with VarSeq from the very beginning to the very end of the process from VCFs to reports. At the end, I can give you a little more detail about some of the other complementary products around VarSeq, such as VS Pipeline and our VarSeq Viewer, which is a free tool to be able to use the projects created from VarSeq, whether from VS Pipeline or any version of VarSeq that's licensed in creating projects. So if you're not too familiar with Golden Helix, let me give you just a 2-minute background, and it helps really understand why it is that we're focused so heavily on the space of clinical genetics. And this is essentially something that we've had a long historical roots in when we talk about, being an analytics company for genomics. Golden Helix is one of the few bioinformatics genomics companies that kind of goes back into the last century, and we've been through a lot of technological and innovation shifts, as well as process shifts from moving from a research where genetics was almost entirely done at a research level to where it is now being applied with great effect in a clinical setting. We were founded in '98. One of our initial key founding partners was GlaxoSmithKline. And as I mentioned, we've been focused through this transition and still provide a lot of support services for the research community. We have over 900 peer-reviewed publications with our tools. Those tools include our research platform, SNP and Variation Suite, our clinical platform for variant annotation and interpretation, VarSeq, and our free visualization platform, Genome Browse, which is very loved, highly downloaded, and that technology is available in SVS, SNP and Variation Suite, and VarSeq. And we might even have time to look a little bit at that today. So that's who we are, and as I said, we've been providing services to and software solutions to customers throughout the spectrum, from small labs to large labs, from individual researchers to large academic institutions, on the government side, on the industry side, etc. And this is really the way in which we've been able to kind of adapt and survive in a very fastly innovative field, is that we are more than just a software company. We focus entirely on the ability to grow a community and learn from that community and put that knowledge back into our products, and to put out things like blog posts and ebooks, and as we're adapting very quickly to the rapidly changing market and technology and bioinformatics standards, we're always updating and being very public about that, our current thoughts and processes and products. And so when you get a SVS or a VarSeq or a Golden Helix solution, you're not just getting a piece of software, you're getting this team that we have built here over the years, and we are there to help you and learn from you, and also customize and put that back into the product. So everything we've built had to be very flexible to be able to keep up with the changes, and you'll see that with VarSeq too. This is not a black box, and we can very easily handle the very individualized nature of a lot of the things we'll be talking about today, like setting up gene panel tests and configuring reports. So with that background in mind, let's also wanted to note that I love taking questions, but let's hold those to the end. But feel free to start asking them using this little pane of your GoToMeeting interface. Those questions that are in there, we will try to respond to. I'll try to save a couple minutes at the end of this. So that's a little background about us, and let's set the context of where VS Reports fits into, and VarSeq fits into this new world of NGS-based gene testing. And I say new because this has obviously been around for years, but I think now is almost the watershed moment is when it becomes economical, and when you can, through reimbursements and through institutional support, efficiently provide these services at a clinical level and be able to have good turnaround and good quality and affordability of the tests so that they can make a strong clinical impact. Because without those things, you're still in sort of the research land, and there's certainly still a lot of work in that context. But what we're trying to talk about today is what it takes to be a viable clinical test. And the argument I'd like to make is that all these steps here that are part of the from cells to reports workflow, a lot of these have now been well characterized as much as any other lab test, in the sense that there are vendors that provide very good kits for doing things like extracting DNA, for selecting down to a targeted panel. So those panels can be hereditary focused, looking for a hereditary diagnostic. They might be also carrier status screening focused or predictive of things like lifetime risk of breast cancer, or they might be focused on characterizing mutations in cancer cells to help inform the targeted therapy of those cancer cells. A lot of solutions out there, and depending on which ones make sense to implement, there are small tests and there's large tests, and the nice thing is you can kind of do something very specific to the institution or the lab needs that you are serving, and there's a lot to go off of with these vendor solutions. And just like any other test, these are well characterized. They have R&D samples or QC samples you can put through them. Get your lab or your targeted panel selected. That allows you to get down to pieces of DNA that are going to be focused on those specific genes or those specific exons of those genes. Then that gets loaded up onto a sequencer, such as a MySeq from Illumina, or the NextSeq, or from IonTorrent, you get the PGM and the Proton. These desktop sequencers have built-in bioinformatic workflows, so if you know you're doing a hereditary gene panel, you just specify that workflow. If you're doing a cancer gene panel, you specify that workflow. That gets you all the way through the sequencing. The onboard informatics will do things like the alignment and variant calling. Again, all this can be set up by any lab that knows how to prepare and run new types of tests. Now, the next step, though, is where it becomes more open-ended, is once you get a VCF file and a BAM file out, how do you efficiently process that data and get it to the point where it can go into a meaningful report? And this is where the hands-on time of a human is absolutely critical and should be optimized for to still make the test viable as a clinical product. So making things like the interpretation platform to do these annotating, filtering, interpreting, and reporting steps has to be a very rich experience that can be integrated into the lab process as automated as possible. And essentially you are optimizing for the time of the person. So if that highly skilled individual can glean as much as possible from their user interface as possible, and then input and capture that knowledge that they are able to apply their domain expertise to, then it can be a viable test. And this is really the problem we built VarSeq to solve. And so again, around that user interface, it has to be simple from the perspective of, can I get all the information I need intuitively? Obviously, it needs to be flexible because every test is different. And that's sort of the process in which you configure all of those choices that are test specific. And it needs to scale because we'd want this to handle not just individual gene tests, multi-gene tests, large-scale, hundreds of gene panels, but up to exomes and genomes. And in fact, we've built VarSeq to do that. We launched it the third quarter of last year. It has all of these features, including our rich visualization engine, and now with VS Pipeline, which I'll talk about at the end, a command line interface. In fact, I'll talk about it in the context here. So this is now what we're able to provide as a clinical suite for this final and important step. So we have VarSeq, which can handle gene panels, whole genomes, has this concept of repeatable workflows, which we'll talk about more in after when we start to dive into what it takes to create a test. We support multiple samples, and we can even make variant assessments from within VarSeq and capture that outside of a project. This VS Pipeline product that we've launched just recently allows that linking between what comes out of the sequencer to utilize VarSeq and do some steps automatically, such as preparing projects that are running the VarSeq workflows, and allows you to support that high throughput scenario. So that's sort of at the very beginning, linking it to your sequencer, and at the very end, linking it to your downstream is this new tool we're launching today, VS Reports, that essentially with one click gets you that final report that integrates all of that tertiary analysis. And we're modeled off of ACMG guidelines. This can be set up to handle hereditary tests and cancer tests, etc. And another thing we'll mention going forward is that we've also integrated Omen as bundled with VS Reports, and this is up-to-date monthly snapshots of this critical database of genotype and phenotype associations and all the literature and references, rich information you get there. So that's the background about what VS Reports is and how it fits into the existing VarSeq suite. And let's go ahead and demonstrate what this looks like from the perspective of the lab tech or the MD or the pathologist who is running through and looking at projects that come off of either a VS Pipeline automated, or they've created a project using an existing VarSeq project template. So I'm going to open up this project, and this could have been one created with VS Pipeline. It could have been one that you did a create new project and then specified the inputs manually. But this is essentially a customized project defining, the workflow steps that have already been validated and now put into production. And this is essentially what then a person doing the interpretation has to work with. This is their dashboard. And you can see right away we have at the top the current sample that we're selecting, and this is a filter chain describing the filters that were defined, the thresholds and the annotations defined during the R&D phase of the test, and now it's in production. We're filtering down to 4 variants. These 4 variants, you can see them at a variant level here, including lots of annotations, which genes they're in, their CDOT and PDOT, number of annotations we have applied here, allelic frequencies from different sources, and ClinVar, etc. And also that extra Omen information that I mentioned, including information about the inheritance model of various disorders that we have here. So that's a great resource for understanding how this gene interacts with phenotypes. So the types of QC you might do immediately is looking at things like the read depth and information from the VCF file, and we can turn on things like genotype quality, etc. So variant level details. As I click on each variant, I get that information down here. We also might look at sample level information. So I'm going to switch on over to a samples view of this project. So switching my table view to samples, I actually have computed coverage statistics. Again, a new feature we added in just our recent release to support these clinical workflows. These are statistics over the target regions for this. In this case, we have a cardio gene panel focused on about 200 genes, and our mean read depth here is 300 and 200 for this sample. And you can see which percent of that total target region is covered by various thresholds. So up to 99.9% is covered at 20x, etc. And we can actually look at those regions individually. So this is for this current sample. Here's all those target regions, and you can see they have names similar to the gene and the region that they're in, and you can look at their minimum depth, etc., and do some QC level information here. So if we're happy with these samples and how they're covered, and we don't have to fail them and rerun them through our tests, we can now decide which of these 4 variants, if not all of them, we want to put in our report. And to do that, I'm going to open up our Reports tab. This is, again, the new feature we're talking about today, VS Reports. And what this has is a report. We call these report templates selected here, and you can have a different template for different tests that you may have in your lab. It can be linked to your project and your workflow, so if you customize one for different tests, you can be running different tests throughout the day. And it pulls in information about the sample that you're currently looking at, and we can have a lot of this auto-filled in from things that are computed from the project, such as these target regions. These are actually the coverage regions, read depth and panel coverage that we just computed and we're just looking at for the sample. And you can even pull in some other information if you, on import or through VS Pipeline's automation, applied extra field information to the sample level. So in this case, we need to then, and this is the critical interface of what the hands-on human experience is saying, which of these variants do I put in my report, and what's the final result? Is it a negative finding? No variants conclusively supported it. Noticed we can actually have report components be responsive to the input here. So let's say we find this to be a primary finding. And we're going to select our red check boxes go into our primary findings section, and our green check boxes here go into our incidental findings. And this gets rendered into the report. And we can say by default, let's set all of these 4 variants to our primary findings. That just checks all these conveniently. Notice what happened automatically down here was that we just filled in these 4 variants into our list, and it auto-fills in information about that variant into its interpretation. This is very editable, and you can change the final classification. So we might do things like look at some of the extra annotations we have here. We didn't filter on these, but we have some functional predictions. These predictions are from things like SIFT and polyphen, these algorithms that say whether they, in a computational biology sense, believe that this missense mutation alters the protein function. In this case, we have 5 different tools here, and 5 out of 5 predicted these as tolerated or not damaging. So let's take these out of our report. So by clicking on that box, I basically remove it from this list. But I might want to add them to my incidental findings section. So let's go ahead and change that and add them there. That adds them to this list of green variants which go down here. So now let's say I'm happy with this. I might also change this final classification to likely pathogenic, or however you may find it, looking at the other annotations. Let's go ahead and with one click get our rendered report. And again, this has information about the sample, the coverage information, our findings, our primary results. For each primary result, we break out and have that rich set of annotations. This was all pulled in from Omen as well as our other annotations in our project. So things like the frequency information from our annotation source. All these hyperlinks can be followed through this. If we're looking at this from a web interface perspective, here's our incidental findings and our background information and our references. And we can go ahead and save this out as a PDF. So this is our final report for 7.8 Rep 1, and have that as a final deliverable, or we could stick with the HTML version with the hyperlinks, etc. So in fact, the hyperlinks, yeah, so that allows you to, and really the customizability of this is something we'll get into in the future and the rest of this webcast, but there's a lot of ways that you can output them. So I'm going to go ahead and close this project because we're going to come back to VarSeq as we talk about the other sort of role of a clinical lab is in creating, defining, and putting into production or designing the R&D process that eventually leads to production of a new test. And we'll come back to VarSeq to do that in just a second. Let's go back to our slides. All right, so that's our demonstration of what VS reports look like. And so now let's talk about what the process is, the R&D process is to design a new test. So getting up and running, VarSeq is been built from the ground up to have the flexibility to handle every test's sort of intricate needs. So the real core concept here is what we call project templates. Everything in that project that I was just in was set by the project template that was put in production for that test. And we can go ahead and define a new workflow by creating a new project and import some representable sample. This is often maybe a spiked-in sample or a sample from, say, Horizon Diagnostics that has cataloged variants that need to be shown that you can detect them with a good quality and high certainty, or at least classify your sensitivity and specificity so that you can be FDA and CLIA validated. And then you'll go through and tune those thresholds to match those expected sensitivity and specificity by looking at the inherent VCF fields. We import every single field in those VCF files. Quite a few of them are standard, things like read depth, genotype quality. Some of them might be specific to your pipeline that have good information for weeding out false positives, things like, you know, some sort of statistic on the forward versus reverse strand bias and things like that. So adding in public annotations, and I'll talk about public annotations in a second, is critical to be able to really get from just a set of high-quality variants to variants that are actually meaningful in the context of the patient's phenotype. And being able to tune those filters to get to that level. I also should mention that if you don't have what you, if you can't find what you're looking for in our public catalog, any standard text file containing genomic coordinates, bed files, VCF files, you can convert those into your annotations that are put into your test and locked down, etc. Including things like HGMD. If you have the offline version of HGMD, you can convert that and add that to the project as an annotation. So all those things are saved, all of those settings, all those algorithms, all those versions of all those annotation sources, and you can document and version that template, put it into production, and then on a new run of data, you just focus on what we did right now, which is that final interpretation step, exporting data, filling in reports. All right. So again, annotations are really key. We spend an enormous amount of time keeping up and cleaning up the public sources that are out there and making them essentially optimized for annotating and filtering workflows. And we're also very proud to be adding Omen as a first-class add-on annotation source. You can add it onto your existing VarSeq license, and it's bundled with your clinical reports VarSeq add-on. And this has very rich and up-to-date hyperlinks back into Omen, as well as lots of very useful, from a both filtering and reporting perspective, descriptions about those research studies and clinical links between phenotypes and genotypes. Finally, as you get all this information into your project, we're locking down to a specific version of ClinVar. We get all this data offline. You're never sending your variants from your lab to a cloud server and having to be concerned about the privacy concerns of having that data transmitted back and forth. This all stays locally to your institution. We get a copy of these databases downloaded, it happens automatically, but into your local install of VarSeq, and then it can be annotated and used against your data. So that's what we capture when we save a project, is all of these settings, including you can start with our starter templates, which we'll demonstrate here in a bit, which have a number of settings sort of at a very good default depending on your workflow. If you're doing tumor normal, we have settings for that. If you're doing hereditary, we have settings for that. If you're doing cancer, we have settings for that. And then you can tweak to your heart's consent and then create a template that goes into production. Also supporting clinical needs, we have this whole thing logged. Every action, whether automated or done interactively, is logging when it was done, the logged in user, and the details of the action, including things like which versions of which annotation sources were used, etc. So one note on VS Reports is that we built this with an eye towards customizability. So what you saw was essentially me just running the report because it had already been customized to that test. But what we'll go through now is the process that you can do some standard customization, such as changing this header information up here and the final sort of disclaimer and test information down here. If you want to change substantially the layout or even the inputs to the report, you know, instead of asking for gender, or you might be having other sort of IDs being tracked, additional IDs, changing the types of information we're pulling from the project at a sample level, at a variant level. all of that is actually customizable through a programmatic interface. The reports are rendered with JavaScript, and if you have folks on staff who are comfortable building basically internal web tools, a lot of labs do, they build their pipelines based on these, they would be very comfortable customizing reports. But if that's not where you want to spend your time, again, with our solutions is a whole lot of customizability and services, and we can configure a service to, say, build 3 different reports where we sit down with you, understand those report requirements, and prototype them and iterate on them until you have a report that looks like and performs the way you need to for your setting. So all this is very customizable, and don't be fooled by the fact that we're just rendering HTML. If you needed this to go into an XML file that got pushed to, say, a EMR or a third-party interpretation service, this is going to be popular for some types of tests like cancer tests, where you want to say, here's the annotated variants with all that information from the VarSeq project as well as information from the lab, and then submit that to a service. We can generate those types of things as well, so just ask us about that. Okay, so let's demonstrate then the process of going and creating a new test template that we would consider as a starting point for going into production. And I'm clearly not going to do every single step that I would want to. That could take weeks, but I think I should be able to show representatively how VarSeq can support the highly customizable nature of preparing a tests for a specific assay, etc. So again, to start building a new workflow, we can work off of an existing starter template, and we have, as I mentioned, a number of these shipped with VarSeq, exome-based, which is going to cover different inheritance models, cancer-based for cancer gene panels, tumor normals for things like tumor normal exomes, etc. And so we're going to start with this hereditary panel, and this is going to be our new cardio test pilot project. And as I hit go on that, I get a dialog to provide a set of samples. And again, I just need a representative sample. So I'm just going to grab this one, and we can come back and validate that this thing works by running a different set of samples once we've built this workflow. So let's grab this sample to start with. And I'm just going to accept all the default settings, and I'm not going to tweak sort of inherent filters or advanced options. So now I'm running that VCF file through, had 14,000 variants, and with a number of annotations applied from our default starter template, our hereditary gene panel workflow starter template, and I can rename this to now be my new cardio workflow. We're going to get down from 1,400 variants to 7 variants. Let's make this big so we can tweak this a little bit. Okay, so remember the types of things that you need to change off the bat are assay-specific parameters from the VCF file. So if I go to this show high dialog, you can see from our imported variant sites a number of annotations that were provided in the VCF file. And from our sample level information, you can see these descriptions. These are all from the VCF files of various different filters and allelic depths and genotype information, etc. The most standard ones that we want to look at are things like read depth and genotype quality. So let's tweak those. If we look at our read depth here and expand out this filter card, I can pull up a histogram of the total read depths for this project. And there's a number of, there's obviously it ranges a whole bit here, but clearly we're looking at most of the reads in this lower region, on the order of less than 100. So let's change the minimum read depth here to just 50. This was not a high read depth targeted panel. So that increased dramatically the number of variants we're looking at from, so we're only throwing away 5 variants that are less than 50. So we may be okay with that from a sensitivity perspective. We may click on those variants to go dive down into them and understand what we think of their quality and their nature, or it could also just revert back to looking at the final set. So we're tweaking things like the inherent properties of the assay. The next thing is to tune what we might annotate against from an external public source. And so from here we have this card is filtering on NHLBI, and we're looking at variants less than 1% or not in NHLBI. That's fine. We might come back to that later, but let's leave that for now. And let's also look at what else we have in our project from our starter template. we have a bunch of information about the functional status of these variants, and clearly we probably want to remove things like synonymous variants, so we're going to filter on the effect and just include only loss of function and missense variants. So that drops in about half the amount of things are going to be investigating, and we can also look at adding a filter based on this frequency, or we already have the frequency field. Let's look at what else we have in here. We have ClinVar. Clicking on any column gives you a description from our public annotations about that column. So clinical significance, this is sort of the all-important classification of that variance in terms of human disease. That's the catalog ClinVar's primary focus. And you can see we don't have any pathogenic variants, but let's go ahead and create in this R&D sample, right? But let's go ahead and create a filter here that's going to be a good one to narrow down to just say pathogenic. And maybe you want to just filter out things that are benign and likely benign and keep everything else, because everything else might be of interest, as well as things that are not in ClinVar might be of interest from an incidental findings perspective. Okay. If we really want to just keep focused on things that have some classification, we can just get down to these two. So indeed, that reduced the search space immensely. We're looking at just 2 variants, but again, this is just an R&D sample. Let's also expand the number of annotations that we have in our project. So I go to Add Annotation. Again, this is using the catalog of public annotations. You can grab any of these and add them at any time. So I want to add, and I'm going to start with the things I have locally here, things like 1,000 genomes. Let's add in those functional predictions like we were looking at in our initial preview. Sometimes those are helpful to weed out variants that are likely to have absolutely no functional impact. Let's also add in the exact catalog. This contains over 60,000 unrelated individuals, and we're going to add in Omen. So remember, all these things that I'm doing, this is what I'm doing when I prepare this test. Once I've settled on these annotations, the person running on a day-to-day basis never has to think about this. This is all part of the built-in project workflow, and now I'm just essentially deciding which of these things would be useful to help that person do their interpretation. So I definitely want Omen, a couple more population catalogs, and functional predictions. Let's go ahead and add all of those. And when VarSeq adds annotations interactively, as well as when we're doing this from a new project template perspective, it can take advantage of the fact that this is a multi-core machine, and so a lot of those annotations will come in very quickly and in the background. And so if I click up here, this little spinning tile, you can see it's computing, and then pretty much finished up to that. So finally, I get these new columns that contain all this information, things like Omen phenotypes and Omen genes. I'm going to probably hide most of these because I don't want this to be a big scrollable list. All that data is there if I need it. I can get to it any time. But let's go ahead and hide all but sort of a couple things that might be useful to help the interpretation process. So I want those functional predictions, and I'm going to bring those up to be right beside my my data. And then I also want, from exact, this is a great frequency catalog, and I might drop a lot of these extra annotations about, what's the count in different subpopulations, and just grab the global alternate allele frequency and move that also up to be right beside NHLBI. So now that I have that cleaned up, you can see my project now has these extra fields. ClinVar, here's that see beside NHLBI's frequency. They match up as you would expect for the most part. They're still trying to grab some concept of a global population. So now I have my project the way I want. I can test sort of what, I might hide a couple more things here. I'm going to hide this column. There we go. All right, so this is nice and tight. I can test things like exports and make sure those all look good. Let's hold off on that until we rerun this template. But let's also configure, make sure we have a good reporting configuration for this, this test. So add again that report tab here. And this is that cardiogene panel, and it's a good start for where we have. You can add new, you can customize and have as many of these as you like, starting with our ACMG template, our cancer template, etc. But I'm going to go ahead and configure a little bit the header and footer information. So with information about the lab, and this is the stuff that's configurable. Once you configure it once, it's permanent. So again, you don't have to touch this on a week-by-week basis. But that logo is customizable. I can say, give me some different logo for my lab, so it looks more like that. And then this is really a different lab. This is Beaker Genetics. So I have a different lab configuration there. So now I can have all that prepared, and I can do a quick test to see that I'm getting a reasonable report off of that. If I'm happy with all that, I'm just going to say that's good. I just want to keep the focus on this as people get started. And again, I can tweak other information about different tables. I could have a table focused on a gene level view versus a variant level view, a sample level view for things like adding in those coverage statistics, which I'm not going to add in now for the sake of time, but it's also something you could put into your workflow. So I'm happy with my prototype, and let's go ahead and move this to production. So I'm going to go ahead and save this as a project template, and this is my new cardio test production V1, and save that template. And again, everything about this is now going to be applied to a new set of samples. So let's do a quick test of that and close out of this project and create a new project. And where's my test? It's the new cardio V1. There it is, new cardio test V1. And so on a week-to-week basis, again, you can actually use this template in an automated fashion by leveraging our VS pipeline, which can run as part of your automated bioinformatics pipeline. After those VCFs are created, you can take this template and produce a project that contains one or more samples. But we could also do this interactively. If we don't have a high throughput environment, that might be just fine. And we could say, here's this week 1 batch of samples for the new cardio test, and we'll create a project that represents those set of samples and add in from the same folder. Let's grab 1, 2, and 3, and again, you could have any number of samples. You can have 100 samples per project. It's really just about what the batch-oriented nature of your work is, and I'm going to just again go through the default settings here. Okay, so now you can see those annotations running. Well, the first step was just to import the variants, so my variants get imported. There's 34,000 unique ones here. Annotations are running, very quickly filling in the information from all those sources that we specified. Those filters are running and being updated. The filters are now done. We're waiting for some more annotations to fill in, but all of the annotations that were required for filtering are done. And we can start the interpretation process. And we can look at the variants for this sample. Oh, actually, there's this one remaining filter is still running here. This is the annotating against our genes, which produce all that critical information about how a variant interacts with the gene, whether it's the sequence ontology and its HGVS notation, etc. So now as I click on a row, I get that rich detailed information down here. I can look at the sequence ontology, HGVS, C dot, and P dot, our annotations for this variant from our different catalogs, and ClinVar's annotation. All these hyperlinks can be opened up into browsers. I can actually dock these browsers into this window. And this same browser can be used for investigating interpretations or the hyperlinks from your table. They will also be used to render and preview those reports. So before I jump into that, let's go ahead and also test exporting. I might be happy with this. I might do my interpretation, set these as my primary findings, and run an export to Excel and grab these two tables and just have an OK on that, and we'll create an Excel file based on my sample name. And our Excel export actually grabs every table in your project as a different tab. So if you had coverage statistics or coverage regions here, they would be here as well. And all these hyperlinks from our project, we try to preserve the formatting and the hyperlinks as well. So here's that ClinVar hyperlink that we just clicked on in the project popping up, in an external web browser. So there's various different, deliverables from a clinical test, including being able to archive or send on information, including all these rich annotations, and it even includes the status of that primary finding, the fact that this was our primary finding. We set that as true as a user. So now we have our filtered results, and we're testing this, and let's test that we can get our report generated as well. So we're looking at our cardio gene panel report, and we're setting our primary findings to be the set of red variants here. and we get those auto-filled in, and we can look at the results, and it's got our customized beaker genetics as well as the 2 variants from this set of samples, etc. So there we go. We have a lot of filled information from the annotations that we all ran. All this was the choices we made when customizing the panel. That can be locked down. One thing I forgot to demonstrate was actually while designing this panel, you can lock down this filter process, and then when you save that project as a template, you can protect that lock status so that this project, which was created from that template, would have this locked and not allow it to be unlocked. So again, another feature requested by some labs to have the certainty that the filters that they configured are run to get these final tables that they might export, and they weren't messed with by accident or otherwise. So we can have that in there as well. So to bring it back to the big picture, I mentioned VS Pipeline because it sort of is a, once you understand VarSeq, it makes a lot of intuitive sense why you might want to automate that one manual step I did of saying create a project, set these VCF files with this template, and then do a bunch of steps, and it computes that project. Now it only took, in this case, you can see it was in live computation 30 seconds or a minute to finish all those annotations. Larger gene panels, high throughput environments, or just removing human error, you might want to automate all that. And that's what you can do with VS Pipeline. So that first set of steps we did where we were defining the project, you still do by hand. Once you're ready to move into production, you can put that template into an automated step right after the VCFs are generated, and then once those templates are created and those samples are imported, you can actually run exports and prepare those reports and do a number of steps with VS Pipeline. And it's designed to be integrated with your command line interface, and it has a well-documented structure for constructing the set of steps that you want to perform. But in basic form, it's create a project, set your input files, such as your VCF files from your batch, and let it run, and then define any exports that you want to run. And the project itself may be the final result, because those projects can be viewed by anybody who has VarSeq, and in fact it can be viewed by anybody who has VarSeq Viewer, which you can get for free from our website. It essentially is the version of VarSeq that doesn't allow you to add new annotations or construct new projects or define new workflows, but look at an existing project and still be able to do exports and have that sort of investigative feel to it, so you can still interact with the data and see what's going on, and then run your own exports, and essentially not do the interpretation work, but the sort of just a viewer level view of projects. Yeah, it's basically read-only projects. So that's it. And if you have more questions that we don't get to today, always feel free to contact us, info@goldenhelix.com. And if you want to see VS Reports handle your own workflow, we are setting up some demonstrations for that, and I'm sure Cheryl will mention next steps on that as well. So with that, I want to leave plenty of time for some questions, and we should be good to go. Yeah.
Thanks, Gabe. Just a couple of quick announcements. I see the questions rolling in. Feel free to continue with those. I will, of course, have the webcast recording and slides up on our website tomorrow, more than likely. And I will email those directly to you, so look for those in your inbox. Our next webcast is coming up on October 14th. That will be presented by our guest, Dr. Sergey Kornilov of the Child Study Center at Yale University's School of Medicine. He will focus on the genetic basis of developmental language disorder. That's all for me, so let's go ahead and get to some of those questions.
Yeah, these all look good. You could probably just start at the top, but I'm going
to start at the top, Gabe.
Yeah. Okay.
Could you discuss further how the default text is created for the variant interpretations on your reports?
Yeah, that's a great question, because we can leverage any information that is in that project. So what we're doing right now is having reports depend on Omen, so it gets bundled with Omen, and you can rely on that information being available. And we also opportunistically grab other sources such as 1000 Genomes Exact, but this is customizable. And ultimately, if you wanted those auto-generated interpretations to contain and again, these are just starting points, these interpretations, but to contain some of your own content, etc., this is all something that you can tweak. And again, these are just starting points because if you wanted to, you can leave that auto-generated information and say, this has also been linked to another family by this lab or whatever. So your own interpretation or your own evidence by of that variant can be updated from that auto interpretation and be inserted into the report as well. So we basically did a set of auto generations based on what's in your project to start with, and it's customizable from there.
Are there any plans to add a CAD score annotation database to VarSeq?
So we're looking into that, and it's basically going to be driven by user demand. There has been a number of functional kind of ranking scores, like these all-encompassing bioinformatics scores out there, and I'm not quite sure if one has a discrete advantage over the other, but feel free to contact us if you're interested in that, or if you have your own license, etc.
Have you updated your article on the state of NGS variant calling? Don't panic.
Yeah, so there's a number of sort of very popular blog posts that I wrote as I was kind of, getting inaugurated into the field of NGS bioinformatics and understanding a lot of the gory details, and kind of as I was learning, putting those out. At the same time, a lot of these are still very current, very up to date. So the Hitchhiker's Guide to Next Generation Sequencing, State of NGS Variant Calling. But I would say that for the most part, from a clinical lab perspective, the validation work is less about worrying about these esoteric edge cases. These edge cases are often not what you're going to be fighting with. And in fact, that's often what the vendors are having done for you, is to avoid all that. They've already tuned their NGS callers, bundled with these desktop sequencers to be handling the edge cases of their platform. So Ion Torrent, tweaks their callers to handle some of their sequencing specific artifacts, and Illumina does the same, and you're not going to have to worry about that type of work. You're going to have to worry about more about, given the quality of DNA, the sequencing depth, what can I expect for a sensitivity and specificity. So a lot of this is still relevant if you're in the frontier of research genetics and you are calling whole genome variants or large structural variants or more complex variants into these scarier regions of the genome that don't have a lot of unique sequence. But for the purposes of NGS gene panel testing, a lot of that's just pretty much handled for you.
Tongue-tied. How frequently are those annotation sources updated? Is it automatic?
It is by our curation team. Things change, so we can't just put everything on an auto script. So ClinVar we update monthly, but ClinVar itself changes their schema and their descriptions and their fields very frequently as well. So there's a hands-on process in which we curate that data and update it monthly for you and match their monthly cycle. Similarly, we'll do the same with Omen. And other annotation sources we update as they are released, since they're not often on a schedule. So things like these functional predictions from DB and NSFP, these are updated as soon as they release a new version. They often are changing things, and we have to keep up with those changes, and then we curate those for you and put them in a very usable package and do things like put these ranking or these voting algorithms in the front of these 5 scores to help usefully assimilate that data.
Is there any way to customize a report to put variants in different categories other than primary incidental? For example, to classify variants as known, likely pathogenic, or VUS?
Yeah, absolutely. So both the things like these categories themselves, as well as these groupings that you might have a different level of detail for, say, the primary and the incidental. These are all extremely customizable. In fact, everything about this layout is defined by what I mentioned is the template programmatic engine, which is basically a JavaScript file. So if you are comfortable in that land of building custom sort of web app type stuff yourself, feel free to use our documentation and do that. Otherwise, if you want to change the structure significantly, we provide a service to do that for you, and we can bundle that in when you get your setup of VarSeq and go through that as an iterative process.
Can VarSeq detect highly variable length but specific insertions like FLT3 TD and call it as an ITD based on its exonic location?
Right, so the highly variable length polymorphisms are, it's not really VarSeq's the issue, it's that NGS and variant callers themselves have a very tricky time describing these in a uniform and useful way. That said, if your NGS pipeline calls that variant in a meaningful way, there's definitely ways to handle those in VarSeqs, but I would certainly say you're a little bit on the frontier of standard NGS gene panel testing if that's something you're trying to get into production.
How is patient information entered into the report? Can it be queried from a database or imported from an Excel file?
Yes, so this information here is being imported based on what I have sort of as a default set of fields from my sample. If you, on the import process or using VS Pipeline's automated capabilities, added extra sample level fields, and those could be something like in VS Pipeline, you might integrate that with a script that calls, looks up the sample and hits up a database and pulls it from a LIMS, and then adds a bunch of fields, such as these, to the project. And if they're in the project, they can be pulled in to the report. So in your lab setup, you can automate and push the data through in a way necessary, especially with VS Pipeline as your engine.
Is VS Reports a separate license to VarSeq?
Yes, VS Reports, like VS Pipeline, is its own standalone license. And again, if you are setting up a lab, we have bundles that will include things like 7 licenses of VarSeqs plus reports plus pipelines, and all that at a very highly valued discount rate, so that it makes a lot of sense to get you going and have all those components to really set up a lab properly to be efficient. If you need to just do a quick, if you have a smaller operation, you just want a standalone VarSeq license, you can get a standalone VarSeq license. You can add reports as necessary if you grow to the point of needing it, etc.
I'll add to that we do have some great intro offers to VS Reports right now as well, and you can certainly get in touch with us or pop onto our website to request pricing or request a trial, and we can have an account manager discuss those options with you. Next question, can information entered into the user-defined database be used in the VS report?
Yes, so we have, and I didn't really touch on this, but we have a variant assessment database integrated into VarSeq. So if you need the ability to classify variants and their pathogenicity outside of individual projects and sort of have that built up over time, so you have essentially your lab's collective assessments. So here's a gene panel database, and you can have, again, this is very customizable. We touched on this on a previous webinar if you want to look at that. This information, including these classifications that you may set on this variant, can be pulled into reports. So that lab-specific classification can be automatically filled in, and it can actually change what this default value is here. So those can be sort of linked up in the sense that if you've classified a variant as pathogenic, you can set it default as pathogenic. Otherwise, maybe you set it as unknown significance as a default. Again, this is part of just defining that lab workflow, how you want to plumb your data through, and kind of how you want to capture information ongoing in a way that it informs future projects.
Is Omen included in VS Reports, or is it an additional cost?
No, so Omen is an add-on annotation source, but if you are getting VS Reports, that is inherently bundled in with that add-on as well. If you need it and you don't need VS Reports, you can also do that as a separate add-on. I think that's about it. Go ahead.
Sorry.
No, go ahead.
You mentioned that there were report templates. Can you expand on that a little bit?
Yeah, and this is essentially what I mean when I say customize, how these things are auto-filled and even which categories are in here, which sections. All that is defined up here in this cardio gene panel template. This is something that resides outside of the project and can be configured, and there's some configuration you can do by this interface that we have here with a set of standard tweakable parameters, and the rest can be configured programmatically. And once you define that, or you work with us as a service and we can help create that and work to match your requirements for that, that can be locked down and put into your production. And again, you could have one of these designed for each of the reports that you need to produce. And so obviously a different panel, you're going to have a different set of disclaimers. You might have everything else be the same, and all you tweak is the disclaimer on the bottom. That's easy to do and clone those, and it's basically a folder with a bunch of configuration files that you just copy and paste, etc. So it's a very customizable per lab process.
Are the reports charged per sign-out or on a yearly license?
Well, that's a very good question. So everything in VarSeq is not based on a per sample or sign out basis. It is a subscription model, so you can use this. It's really your business. We don't want to be involved or try to involve ourselves on a per click basis. So the yearly license that you get, or you can actually get multi-year licenses if you want to lock in better rates or have that certainty on the price structure over time, covers things like VS Reports when you add that as an add-in to your license, and there's no further per user or per sign-out level monitoring. And in fact, all this happens on your machine, so we're not actually capturing or knowing anything about your volume.
Does VarSeq provide coverage statistics?
Yeah, and I didn't have a chance to touch on that because it's a recent feature, but it is very useful and probably critical for in a standard gene panel environment. So I'm going to go ahead and close out of this project and go back to that project that I started with. And this was, again, a fully computed project that we were doing some interpretation on. And I mentioned this view where you have these coverage computations done on a per region basis, and then this view where we have those rolled up at a sample level. So this is for the 2 samples in my project, their average coverage totally over those regions, etc. And all this is integrated again with our visualization. So now is a good time to demonstrate that. If I double click on this, I could zoom to that region, and by default we just have our genes here, but let's go ahead and add in the sorry, I have the wrong folder selected. Go back to this folder. Yep, and grab these samples. Oh, I guess it's in this folder here. Okay, so if I have my BAM files that I want to look at, in this case, it's this guy, as well as the, we might have the target regions themselves. In that case, it's this cardio gene panel design. And I brought in the variants, not the BAM files. Bring back this, go back to my browse, go back to the sample. Oops, I want this BAM file. Thank you. So these coverage regions you can introspect by simply navigating them in Genome Browse. And as well as, of course, you can do this at a variant level. So you can do the QC if there's a need to sort of introspect. This is the region. This is its coverage. You can find regions that have low coverage by sorting, etc., or filtering. You can look for poorly defined regions, and okay, so yeah, this has a little less coverage here, and whether I'm okay with that, I'm in the intron of this gene. And similarly, you can have those types of click and navigate views on the variant level. If you're doing some QC at the variant level, here's that A to G mutation very clearly defined in the BAM file, in the VCF, and in the where we are in the gene in the middle of this exon, etc. So coverage statistics are in VarSeq. They can be part of your workflow. They can be computed with VS Pipeline. They take this BAM file and this region file and essentially give you the statistics over this region of your average, minimum, and maximum read depths, and help you define regions essentially that are potential false negatives where you would not be able to detect a potentially significant variant from a clinical perspective, and then help you decide, oh, I might have to fail the sample because of that.
Okay.
All right.
This seems like a good place to wrap.
Yeah, absolutely.
Awesome. Thanks so much, Gabe. Great webcast. There are some questions that haven't been answered. We will make sure that you get answers to your questions. Either I will be in touch, or I will have an account manager get in touch with you. As I mentioned, I will shoot you the recording link and slides tomorrow. As well as if you're interested in taking a deeper look at VS Reports, we do have some great intro offers. You can email us directly or pop onto our website and request pricing, and we will get in touch with you. I hope everyone has a great day, and thanks so much for joining us.
All right. Thanks, everyone.